Okay, I'm going to go ahead and get started. First thing is to say a few words about the homework that I just posted for Module 11. The main reason it didn't come out last week is that I was really trying to make the single cell and the metagenomics as similar as possible, so it doesn't really matter which one you picked. You have to do about the same kinds of things. Chime just wasn't gonna let that be a thing. So I think in the end now they're roughly equivalent levels of work, but they're kind of different in terms of the kind of work you'll have to do depending on which one you pick. So the single cell RNA-seq tutorial will require you to apply the classifier. So you have to do more modification to the notebook. So this one is a little bit more read the code and apply what you know from the classifiers to the ScanPy tutorial. So it's sort of building off the end of the ScanPy tutorial. The metagenomics one has a lot more manipulate the plots and interpret what you see. And again, you only have to do one of them for full credit, but if you do both, you get extra credit. It's 2% toward the end of the semester grade if you do both, and that is explained in the assignment itself. So in the module, there are 2 assignments there. They both, if you go to it, what you'll do is download that Word document, fill it out, and then re-upload it. And so that bit about only needing to do one of them is also included there in the instructions. I talked a little bit about why Chime is the way it is, decentralized development, in terms of what the main problems tend to be. And it's funny reading some of the documentation and the tutorials. I probably tried to do half a dozen different tutorials with Chime. They are all broken. Like, that decentralization that they tout as a feature is a bug. So that is all to say, the reason, the place where it is broken for us, like if you try to use the latest methods, and this gets to some of the questions that you're asked to interpret, right? This is the workflow for Chime. And I kind of want to make sure that I asked this question on Thursday, but just to make sure people are understanding where this fits in the context of machine learning and classifiers and what the objective of that module was. This part here, so our notebooks run all the way through Dada. So that does the clustering and the denoising. That is analogous to getting through the PCA in the single cell analysis, where it will generate clusters. In metagenomics, those clusters are the clusters of similar sequence that in principle correspond to some taxonomic level of microbe. Where our notebook gets broken is in once you have those representative sets of sequences, those sequence clusters, going to identify what the taxonomy is. So that is where there is a classifier, and it's questions in the homework will ask you to go figure out. It's in the documentation for the tutorial, so I'm not asking you to like actually go do research, at least for this part. But understand what the classifier is. And so that's the tie-in, because ultimately that's what I wanted the objective of the metagenomics part of the assignment to be. So once you have that taxonomy, and you can then do all kinds of things about differential abundance based on which kinds of sample it came from, and there are a bunch of cool visualizations you can do. Now, for the homework, what you have to do is go through that page, the gut soil axis tutorial page, and instead of running all those samples yourself, you just have to visualize their computed ones. So in case that didn't make sense, where can I go here? So this link at the top, it's just not a link. Just to right. So because there are important parts broken, you can just go through this, and hopefully the questions in the Word document are sequential with the sequence in the tutorial here. And so several of these like QZV files, you can click view on those and it takes you automatically to the viewer of that output. And so that's what you have to rely on to answer the questions in the homework. Questions? Does that make sense, I hope? Maybe not in the abstract, but hopefully once you take a look at the Word document, it will come together. Mm-hmm. They're in the Chime, they're in this. So this was the, and it's also that URL is at the top of the homework page. And if you're not into metagenomics, do the single cell one. It's a little bit different kind of homework. I think the application of classifiers is way more clear from the single cell analysis, because we actually sort of did both, and it's just putting them together. Yeah. Okay. So I made the due date for that Monday. That's soft deadline. Okay. It's that homework I think is probably going to take a little bit of effort, more so than some of our previous ones where it was just copy paste the figure at the end of the notebook. Because there are questions in there asking you to really understand what the data are and what you can interpret from the data. And the other reason it's soft is that the rest of the semester is final projects. So today, the objective is, I have it listed as project proposal workshop. I'll turn you loose here after I run through a few slides just to get your brains working on what final projects might look like. So in the Canvas module, there is a PowerPoint that I'm going to go over. So Tuesday lecture for Module 12 is this. So there are 4 components to this final project: the proposal, there'll be a draft report, peer review, and then a final report with a presentation. I'll give you lots more detail and rubrics for all of those on Thursday. Okay. Today is mostly about getting you on your way to that first thing. Like, what am I actually going to do? Okay. So in some ways, that's the hardest part. There are a variety of types of projects that you might do. You can do a reanalysis. This is finding a paper, for example, and reanalyzing their data with an updated method or something like that, or extending their analysis in some way. Those are nice because you should be able to show that you can reproduce sort of a known answer and then how you made it better. You can develop a new pipeline. You can do some of the stuff that we just did in Module 11 with maximum likelihood models. Maximum likelihood. Machine learning. Benchmarking. So does a certain algorithm run faster or give a more reproducible result? So comparing algorithms or quality control approaches. I think. Yeah. I was trying to remember if we used any benchmarking data sets in here. I feel like we have, but it's not coming to me. Whenever somebody develops a new algorithm, there's often a benchmarking paper that comes along with it. For example, if they publish an update to Clustal or one of the multiple sequence aligners, there are benchmarking data sets where they'll run all, like the old version of Clustal and the new version of Clustal, and then compare the results of that to some of the other methods for multiple sequence alignment. That's the kind of thing we're talking about. Building a dashboard. So some of the things like we did with Plotly, showing 3D visualizations and things like that. How ideally these things are, going back to my beating on the drum of look at your data, building a dashboard that makes that easy for the non-bioinformatician, right? This is you as the bioinformatics person making life easy for the rest of your lab. And then there's the hypothesis-driven biology, right? If you're in a lab and you've got data and a question already, knock yourself out. Ideally, this is not a project that is done in isolation from the rest of what you're doing with your life. Especially for the PhD students, use this as an opportunity to make some progress on preliminary data or preliminary analysis. Okay. Questions about types of projects. I have lots of examples coming up of more specific questions and things that you might address. Mm-hmm.
I have some things from my work that I could probably use for a project, but they're like really old, I guess you'd call it ancient DNA kind of stuff, archaeological projects, stuff like that. I don't know how much of a complete genome I may have, like of some bacteria, for example. Would that be not recommended for this kind of thing? Because maybe
no, absolutely. I've actually got that listed in one of the slides later as a ancient DNA. How do you deal with that? Error correction in ancient DNA. Yeah. Or identifying metagenomic stuff in ancient DNA. I don't know what else is in there. Yeah. I'm super excited about what you guys come up with for projects this year, because ChatGPT. Right, it is your superpower in terms of coming up with and exploring the ability to actually carry these projects out. So the next list, a lot of these go along with modules that we've done, and some build off of ideas that we have covered in more or less detail. You can pick one and adapt it, or just, ideally using these as inspiration for your own ideas. So some of you, comparative genomics is big in the biology department here, and so lots of us interested in how genomes evolve, what are the evolutionary forces acting on genomes. So here's a little bit of a laundry list of those kinds of ideas that you might want to do. Anybody know what DNDS is? We didn't talk about molecular evolution that much. What's a DNDS?
Ratio of non-substitution to substitution.
Non-synonymous substitution to synonymous substitution, right. So that is a metric of the action of natural selection. So if the non-synonymous is much higher than the synonymous, that's a clear signal of positive selection. Copy number variation could apply, Café, or is the one that I built or helped build in my postdoc. Lots of people use for structural variation, especially gene duplication and loss. If you have a new genome and you want to compare it or annotate it, those are good projects. Predicting deleterious mutations, that's a sort of functional mutation analysis. And then ortholog inference and gene family evolution. Those are like projects we did, but would expect you to do that on some new data. Microbiomes and metagenomics. I would be okay if somebody built a Docker instance, for example, that actually worked for QIIME. As a sort of not biologically motivated but super useful bioinformatics project right now. But if you've got data, especially, and there's tons of, in the slideshow I gave the human microbiome database slide, but there's lots of data out there for humans specifically, but you could comb the literature to find data for other stuff as well. And there are a couple labs here that are doing that kind of stuff. The Ravenscraft lab. I heard yesterday the Frischkopf lab might be getting into this. The Chang lab has a big component of what they do on this, both soil and human gut microbiomes. RNA-seq projects, single cell or whole, right? So the next one is single cell. Yeah, I don't need to read all of these. These are meant for you to look at and hopefully get the wheels turning.
What do you mean by LLM-assisted annotation?
So if you take a new unannotated genome and you train it on, actually there are training data sets out there, but then it's using the same kind of models that underlie ChatGPT to then annotate, like find what the genes are, what the non-coding functional parts are. This is a whole field now, because genome annotation is the bottleneck in making use of new genomes. We can sequence genomes fast now. Even good, well-assembled genomes can come out fast, but annotating them is still a major bottleneck. And LLMs seem like they're the way to go. And I'm not sure what the frontier is right there at the moment. But if I had the time to pursue that research area, that's what I would be doing. Okay. Anybody who has proteome data, there's a bunch of stuff we didn't talk about protein very much at all. If you don't know what some of the acronyms are, that's probably not an area that you're going to be working in. Population genomics, this is sort of similar to the first slide, evolutionary genomics, but thinking more at the population level and variation across spatial scales. Pipelines, this is one I talked about with metagenomics already, but making some kind of containerized version of a software package that you think would be useful. A huge, like maybe the main employment area for bioinformaticians is you are plugged into a biology lab and asked to make everyone else's life easier. In terms of, like, they can't do the analysis. You are the key to unlock all this biological data that they're collecting. And so one way to do that is push the buttons yourself. The smarter way to do that is make buttons that they can push. Okay? So that's what the pipelines and the reproducibility are. That's creating the button for the biologist to push rather than having to be there to push the button yourself all the time. And so we have a bioinformatics hire coming up. The first candidate will be here a week from now, so on the 10th. So we've got 4 candidates coming in. So if you want to come in and see seminars, I'll post once I have the details for times and locations when those seminars will be. But if you want to know on the academic faculty side what it takes to get a job, those would be really good for you to come listen to. And you'll get an appreciation for what does it mean to have a bioinformatics research program. So this is one of the things the search committee is struggling with, is somebody who uses bioinformatics tools versus somebody who develops bioinformatics tools. And so you'll get to see a range of those kind of talks and those kind of research programs. Pipelines and reproducibility would fall in the category of somebody who uses bioinformatics tools mostly, right? So it's building that container I was talking about, like for Chime, versus somebody who's doing the LLM annotation thing. That would be somebody who's actually developing the tools. Neither one is sort of better or worse. They're just different kinds of research programs. Visualizations and dashboards, I mentioned this a few minutes ago. Would be really cool if somebody did a spatial atlas visualization. Would love to, that's on my project list for a collaboration with one of our new faculty members. Because zebrafish has a spatially located single-cell RNA-seq database. So when you do an experiment on zebrafish brain, we can map the expression of the individual cells in 3D space. And so building visualizations for that is going to be very cool. I'm thinking put the Oculus on and be inside the brain. I'm not joking about that. And then here, like I said, so the last one there, ancient DNA contamination detection. It could also be, how do we analyze ancient DNA, given that it has high error rates? You have to account for the kinds of mutation that happen as DNA degrades over time. Yeah. Okay, so I'm going to turn you loose. The objective for this week is to get on the Google Sheet. Ideally, that takes you to the Google Sheet. So this has a place for your name, your UTA ID, your project title, the biological question you're going to address, the analytical approach. And I'm not asking for a lot of detail in this sheet because you are going to have to turn in a one-page proposal. Okay, this is just to give me an idea. Well, one, to make sure that everybody's on here and everybody's thinking about their proposal, and that this will potentially give me enough just to know, is this going to be feasible? And thinking about, your question, your approach, and your data are very important to that feasibility question. Did that actually switch over? No, it didn't. I'm looking at the sheet, and right, so the Google Sheet has these columns that I was just talking about. And you can work in groups of up to 5 people. I will identify those groups based on having I'm going to sort on the project title column. Okay. And then each member of the group will need to have a specific role, and that's going to come along with an extra reporting step in the final report to explain what each person did. Okay? Any questions? So, yep.
So you are definitely sorting us into groups, or you might not if you're like not aligned with
The groups are self-identifying.
Oh, I thought you.
No, so you can be up to 5, and you get to make those groups yourself.
Oh, you meant on the sheet you're organizing it into groups.
Yeah. So once everybody's, because even if you and the rest of your group decide to go in and put your things, it doesn't guarantee that you'll be next to each other on the sheet. I was just telling you how I'm going to identify the groups.
Oh, okay.
Because I am not assigning them. Yep.
So as a group, we're just doing one topic, or are we blending?
One title. If you like how you split your project, if it's broad enough, like if you have 5 people in your group and you want to build an LLM to do annotation, you could do that on 5 genomes and each person do a different genome, but you're all working on building the LLM yourself, or yourself as a group. Yeah, how you divide the work, I am also not specifying. Partly because that'd be impossible to do generically with so many different potential project types. I don't care whether you stay and do this now in person or you do it offline. Obviously, if you're in the online section, you're doing this all offline, but you're also welcome, if you've got friends in the class, to coalesce on topics. I guess they don't have to be friends. Collaborators.
By when do students need to fill out
I don't have a deadline for this. On Thursday, we're going to talk about the one pager. I'll give you an example for the one pager. I'll give you rubrics for all 4 of the components so that you've got concrete targets for what you're going to turn in. And that one page proposal will be due next week. Okay. So let's go look at, we are mostly on target. So this project proposal says that's for this week, right? So that will actually be due sometime next week. But then 2 weeks from now, right, on the 18th, that week we will have a project status report, and that is going to be peer reviewed. Okay. So those will be distributed randomly by Canvas for everybody to look at and give feedback on. And it's just a mechanism for both you to see what other groups are doing and motivate you to not wait till the end to try to get it all done. Okay. And then final reports, we'll talk about this. Presentations will be short, and we may not be able, this depends on how clustered people are. If we have all groups of 5, then maybe we can do more presentations live. Otherwise, they'll be recorded. And there's a written part of it. But all that detail will come Thursday. Other questions? Yeah.
It's view only.
The worksheet is view only? Thank you. Okay, should be fixed now. Okay, that's it for me. So this is also potentially, if you are interested in having partners but don't know any, you can use the discussion feature of Canvas to say, hey, I've got some data. Anybody want to help analyze it?
So somebody who can like look at the data that I have to make sure it's like usable for the project.
Yeah.
A lot of these old samples they'll have a ton of contamination, a ton of damage. Don't know how much is sequenceable in there, kind of thing. I've sequenced it using Oxford Nanopore sequencing.
Uh-huh.
I don't know the quality of it.
That seems like a good project. You tell me what's the quality of it. But so, it's a good comment though. The rest of the semester, essentially, this is going to act like office hours. So if you've got an issue with your data or your analysis, that's what class time is going to be for the rest of the way. Like Thursday will be my last lecture. After that, it's just me and Adam and Zoe trying to help you guys get accomplished what you set out to do. And it's also open for people watching this later. If you're in the online section and you want to come during the office hours, during class time to get help, definitely do that. And then, of course, our regular office hours are also available for that.